It is my pleasure to welcome previous students, fellow researchers to a symposium recognizing Professor Clark Colton's contributions to our careers as well as to the science of medicine and engineering as a true pioneer in bringing engineering principles to treat serious medical diseases. Just like to recognize Clark. You want to stand up for just a minute, Clark? Just turn around and look at everyone. It's been my privilege over the past couple of months now to work with Clark and with most of you in terms of learning more about what you're doing and the exciting things that have happened before as well as after I was here and spent time in this room that looked a little bit different at that point in time. But it's an exciting time, and I think we have an exciting illumination of the contributions that Clark has led and have been done by many of the people in this room that we'll be sharing throughout the course of the day. So I have the pleasure of beginning by doing a brief introduction of Clark as, again, a pioneer in really applying chemical engineering principles to serious human health diseases. This career almost didn't begin because when he was at Cornell, Clark was thinking more about aerospace as opposed to biomedical engineering. But when he got here, he took Ed Merrill's course and realized that physiology is all just chemical engineering. In his words, jumping down, diffusion is going on at the local level. There's a lot of physical chemistry involved. There's fluid flow, blood flowing through the veins and cardiovascular systems. There is transported mass everywhere, in and out of cells, across many kinds of organelle boundaries. Much of it was chemical engineering. I found it fascinating. And that really began his course in terms of focusing chemical engineering principles on biomedical problems. His advisors were Dr. Ed Merrill and Dr. Ken Smith. For all of those of us who took classes, who studied under Clark, you'll appreciate when Ed Merrill pulled him into his office and said, after studying in the library, researching in the library for 6 months, Clark, you're not writing anything. If I don't have your proposal on my desk back by Labor Day, you've got to find a new advisor. We may have heard similar words ourselves at various times along the way. Dr. Ken Smith became a close collaborator with Clark throughout the course of his career. But over 50 years, Clark pursued applying chemical engineering principles to create major advances that have benefited many patients and across an amazing area of research: diabetes, stem cell therapy, tumor targeting drugs, immune engineering, atherosclerosis, just to name a few of them. I'm going to give a very brief overview of just a couple of these areas to give a sense of how Clark approached these problems over the years and his tenacity in searching for solutions. In diabetes, as one of the examples I'm going to use, the goal was always to achieve normalization of blood glucose levels by providing the right amount of insulin. The mechanisms to do that varied dramatically over the course of Clark's career. His first attempt was using electromechanical systems, developing a sensor, glucose sensor, to control a pump. This was work helped by, throughout all the work I'm going to talk about. In blue show the names of some of the researchers who helped doing PhDs, postdocs, and others working in Clark's lab. But this required development of complex algorithms to be able to control the right amount of insulin at the right time. One of the key discoveries was that the sensor lag could be no more than 15 minutes. Otherwise, you started to lose control. The second area moving in terms of diabetes was to look at curing diabetes by the transplantation of islets of Langerhans. It was well known at the time that you could transplant islets of Langerhans, but they were rejected by the immune system and destroyed quickly thereafter, unless you used immunosuppression, and to be honest, immunosuppression was often worse than diabetes itself. So Clark's concept was to pose an impermeable barrier, a membrane, to prevent rejection while still allowing the nutrients to keep the cells alive. His first attempt at doing this, first of many attempts, was to use an AV shunt and putting the cells in the outside shell where they could be perfused or have be benefiting from the shunt perfusion system, which actually worked in terms of being able, from the diabetes perspective, in terms of being able to control blood glucose levels. The problem was the system clotted which meant it didn't work in terms of long-term health for the patient. The second direction, also in the area of a hybrid artificial pancreas, was to encapsulate the islets in a way that didn't require immunosuppression. Work that I was part of was really the discovery. Just as a note to our AVT, it's shut out. I don't know what we need to do to keep it moving on the screen in front of me, if you could maybe look at that. But really discovering the role of hypoxia as a major effect in terms of regulating the ability of encapsulated islets to be able to function. In fact, oxygen diffusion throughout the process became really one of the key factors that limited the ability to encapsulate islets or transplant them in many different formats over the course of efforts of mine and others as well thereafter. The next attempt became trying to solve this oxygen limitation problems, working with Susie and Gordon Weir, in terms of using microcapsules, thinking if we use small aggregates in small capsules, there'd be the ability to have sufficient oxygen to be able to penetrate and keep the cells alive. The next attempt went to working with scientists at Baxter to develop capsules themselves whose microarchitecture could drive the ingress of new capillaries to provide oxygen as close as possible to the membranes, again, in order to keep the cells alive on the inside of the encapsulated tissue. From there, making a slightly more elaborate approach was saying, well, can we just provide more oxygen to the cells by adding an electrochemical oxygen generator next to the encapsulated tissue, provide a local source of oxygen, which actually worked in terms of being tested in rats, primates, and humans. The challenge was that it was a fairly complicated system and difficult to be able to maintain. So it moved on then to the next attempt, which was really to say, can we treat the cells themselves, develop better islets, better beta cells, increasing the fraction of beta cells versus other cells in the encapsulated tissue, to be able to decrease the need for oxygen and be able to have the cells provide sufficient insulin, again, to control blood glucose. This was work done by Piazana, Pappas, and as well as others, developing the oxygen consumption rate measurement to really be able to quantify the effect of changes they were making in terms of the tissue that was being encapsulated. After that, the concept became, well, maybe we can increase the amount of oxygen coming in by changing what we make the membranes out of, using silicone rubber for membranes to be able to enhance the transport of oxygen into the encapsulated tissue. All total, as I looked at that, go back one on that, there were 7 different approaches that Clark drove with various different students to try to solve the challenge of transplanting islets to cure diabetes, making a lot of progress along the way, each one facing different challenges. One challenge was solved, other challenges came into play. But it shows that, again, the tenacity over 50 years of trying to solve a major problem of type 1 diabetes that so many people in the world suffer from. The second example I wanted to share was that of atherosclerosis in the different areas that Clark has worked on. And atherosclerosis was one of the major efforts throughout Clark's labs for many years going forward. Probably had the most students working on it. Really to develop initially, the goal all along the way was trying to understand what was the cause of atherosclerotic plaque formation and how could that be controlled, whether it was looking at lipid transport, whether it was looking at shear forces, models of blood flow through various vessels, and identified a number of different causes, a number of different theories that were actually formed a very comprehensive theory of the formation of atherosclerotic plaque. Identifying, amongst other things, that looks more on track, yes. A number of experimental techniques that many people in the audience today worked on in terms of analyzing the location and extent of plaque formation, as well as then looking at a variety of mechanisms in terms of understanding why and where the plaque formations occurred. The kinds of questions that were asked are these shown up on the board. Does LDL enter the wall predominantly from one or both sides? Is it driven by diffusion or convection, which is dominant? What about the layer of endothelial cells? How does that play in terms of what happens and where the various plaques form? What's the effect of opening endothelial junctions? Is the uptake uniform or is it patchy? And does that patch, if so, correlate with the formation of the atherosclerotic plaque that later formed? Does LDL bind to tissue components? Is it degraded or modified in the wall? Can a mathematical model help predict where the plaque formation might happen and why it's occurring? So again, out of that, there were major discoveries identifying different forces that drove the formation and location of the different atherosclerotic plaques, whether that's shear stress, whether there's division of cells, whether that's particularly hot spots with macrophages, and velocity profiles to help to compute that from various mathematical models. An overall hypothesis came out of this that basically said that flow and other stimuli cause endothelial cell injury, which then drove much of the underlying, exposed the underlying tissue, leading to local high concentrations of lipid-dense proteins. When this is degraded or modified, it attracts inflammatory macrophages, which then attracts LDL, thereby forming lipid-laden foam cells that really lead to the beginning of the plaque formation. There was one missing link that wasn't discovered until later, and that was the role of oxidized LDL, which contributed to this as well. But overall, it was a very comprehensive understanding of atherosclerotic plaque formation. Sorry, just time-wise, these are important areas, but the presentation that we actually wanted to give here tried to summarize. Yeah, we're on the wrong presentation, so we'll just jump from here on it. The reason I shared these two stories is really to demonstrate how Clark approached the concept that drove his research throughout the years. And that was really to go at, understand it mathematically, understand it from an engineering perspective, understand it from a biological perspective, and then engage students who are excited about pursuing engineering in the body to engage in major experiments and work going forward. In the atherosclerotic plaque area, out of a short 15-year period of time, 12 out of 14 students out of Clark's lab received academic positions going forward. During that same 15-year period, 7 other students coming out of the lab also had academic positions, for a total of 19 academic positions filled from one lab over a 15 period, which may still lie as a record in terms of the number of people coming out of one lab, filling academic positions and teaching many other students, really to pursue the kinds of things that excite us about applying engineering principles to biology to make fundamental advances that hopefully can cure people, ourselves, our children, our neighbors, and others around us. With this, I'd like to come back and say the reason for this symposium is really, Clark, a thank you to you, not only for beginning our careers, but also for enabling us to help the hundreds and thousands of patients through the discoveries that many people have done, driven by their initial work in your lab, really to think about engineering and physiology as the same subject, and working forward to find solutions to cure serious human diseases. So thank you, Clark. Just as a note to everybody, there are packages that contain summaries of a lot of the slides you're going to be seeing in the back of the room that Clark put together. So feel free to take one home with you along the way to really have that as a memento of both your work and Clark's work as well.